Thank you, Cypress. Well, good morning, good afternoon, and good evening to everyone joining us today. My name is Tricia Mischler, and I am the Director of Regulatory Affairs at Mead Johnson Nutrition, which is a member of the Infant Nutrition Science Coalition. I have worked in the pediatric and specialized nutrition space for my entire career, and I am absolutely honored to facilitate the conversation today with some esteemed colleagues in this space. On behalf of the Infant Nutrition Science Coalition, thank you very much for joining the INSC's first Scientific Roundtable in the series. The objective of the Roundtable series is to provide a forum for experts to discuss pertinent scientific questions that support and are parallel to the focus areas of the INSC. Today, our roundtable discussion will focus on approaches to establishing scientific consensus. Before I dive into today's session, I do have just a few housekeeping notes to share. So you can probably see that we are being recorded, and Cypress will be posting that recording to the INSC website in just a few days. Throughout our session today, please feel free to type your questions in the box titled Q&A, not the chat box, but that little box called Q&A. And I will do my very best to get to all of your questions today with our expert panel, but based on timing, we may not have success in that endeavor. So we will try our best. For those of you who are unfamiliar with INSC, we are a tripartite initiative bringing together scientific experts from academia, government, and industry to advance research on human milk and infant nutrition. The INSC aims to improve our understanding of human milk's composition and functional effects, while also exploring how differences between human milk and infant formula may impact infant development. For more information about the INSC and our initiatives, please visit our website at inscoalition.org. I do also want to take a moment to thank the INSC Steering Committee members who helped plan and organize the session: Vanessa Castanga, Paul Hanlon, and Barbara Schneeman. Thank you for your expert guidance and direction on getting us to the roundtable today. For today's session, we've invited 4 expert panelists to share their different perspectives and approaches to establishing scientific consensus. Using a real-world example, we will evaluate how to incorporate principles from these approaches into INSC's approach to reaching consensus on some of the outstanding yet fundamental scientific questions related to infant nutrition. I am going to share my screen now to ground our discussion. We've asked our panelists to reflect on the following question. Cypress, you can see my slides, correct? Okay. All right. So again, we've asked our panelists to reflect on the following question, not to answer the question, but to explore their approach to establishing scientific consensus. And that real-world question that we ask them is, how should a single representative concentration of a component in human milk, including both essential and supplemental nutrients, be chosen? This question is deceptively simple, I agree. But it touches on the heart of considering what process approach can be taken to reach scientific consensus. With that, I have some thought starters to help set the stage for today's discussion. Before we jump into the panelists and their ideas and discussion, I'd like to take a moment to just reflect on the nature of scientific consensus. It's a term we often use, but we rarely pause to unpack it. What does it mean for scientists across disciplines, sectors, and perspectives to agree? Not everyone has to agree, right? Well, consensus is dynamic in that it evolves as new methods or perspectives emerge. And it's also contextual because it depends on the question that we're being asked or the quality of the data that we're dealing with. So, is consensus always possible? And if so, how do we get there? There are many different terms used to describe reaching scientific consensus today, and they are often framed as the level of scientific agreement. We have the weight of evidence, which is based on the totality of available data. It's often used in risk assessments. It emphasizes the balance and integration of diverse data sources, and it may include some conflicting findings, but the trends point towards a consistent conclusion. And then there's significant scientific agreement. Those of us in regulatory are very familiar with this term because it's used by the FDA in evaluating health claims and GRAS determinations. It requires broad agreement among qualified experts based on publicly available evidence. It doesn't suggest that everyone agrees, but there is a high level of confidence in the safety or efficacy of the substance at issue. And it's often studied or supported by multiple studies. And then there's reasonable certainty, which is a legal and regulatory threshold used in safety evaluations, such as food additive petitions. It means there's no significant doubt about the safety under the intended use. It requires a robust data set, often including tox studies and exposure assessments, and it's used to justify regulatory approvals and other public health decisions. Understanding these levels of agreement can help us clarify our expectations and really guide the decision-making process. These levels of scientific agreement matter because they influence and drive policy and decision-making by health authorities, not only in the United States, but around the world. They set a foundation for how to determine which level of scientific agreement is needed for differing decisions, such as nutrient recommendations or claims. And they help with different types of decisions and help determine which framework best suits the consensus building efforts. Again, today's session is not about arriving at a final answer for that question that we posed to our panel. It's about exploring the process, the principles, the tensions, and the tools that scientists use to navigate uncertainty and building that consensus. So let's explore this process with our experts. I'm now going to take the opportunity to introduce our esteemed panelists, and I'm going to keep my introductions for each panelist brief, but you can find everyone's full bio on the website, again, at inscoalition.org. I'm pleased to introduce our first panelist, Dr. Daniele Wikoff. She is Chief Scientific Officer at ToxStrategies, as well as Senior Vice President of the firm's Health Sciences practice. Danielle, I'll hand it over to you.
Wonderful. Thank you. Thank you for the invitation to be part of this discussion today. I think it's a really exciting and it's a great topic. It's certainly a very challenging topic as well. I'm going to go ahead and share. Sorry about this delay there. Okay. Well, thank you. Okay, so as introduced, my formal role is as a Chief Scientific Officer here at ToxStrategies, which is a consulting firm that provides services to both the public and private sector. But my informal title, I like to think about myself as a bit of a methods nut. I really am passionate about the use of evidence-based methodology and its capacity to help solve questions and address aspects like consensus. So I wanted to start with kind of how I think about the thought of consensus in science, and just have a little graphic here thinking about kind of going from divergence to consensus. So going from many different opinions or many different aspects of science to a single opinion or weight of evidence, scientific evidence as it relates to the research question that's been posed. And the way, in my mind, that we achieve that is really based on our knowledge. So as our knowledge increases about a topic, and that can take many different forms, the amount of evidence, the diversity, as our understanding of the biology and the science advances and our ability to really inform these questions, all of these aspects really help to increase our willingness to achieve consensus, to converge and to consent. I do feel like as you grow, as this information also grows, it influences each individual's beliefs and norms. So while you have evidence-based really informing that, every person will have these opinions about the science. A couple examples I thought worth throwing out of where I see our field. One example is with non-animal testing and NAMs, these new approach methods, things like using in vitro data to predict human responses. We're still at the beginning of this, and our knowledge is growing, but we are certainly not at consensus or even convergence on the use of these for regulatory purposes. In the middle, I would put something like behavioral disorders. We are hearing a lot about autism and ADHD and environmental factors and nutritional aspects. And one of the reasons that we are still investigating this is these are complex disorders. We don't have enough knowledge. So we're learning more, but I would put that a little bit in the middle. And then on the right side is where our knowledge about vaccines. I would actually give this as an example as well. There is a large amount of convergence. We maybe don't have consensus as a field. So when I think about achieving consensus, one of the aspects that comes to mind is this discussion of gold standard science. And we've also heard a lot about what gold standard science is from our administration in the US, but really this is a topic globally. And when you look across how this is defined, whether it's in the US or in other geographies, you see a lot of the same types of themes. Reproducible, transparent, collaborative, unbiased, lacks conflict of interest. And I think that's an important stage to set with respect to achieving consensus. So I really see the ability to use evidence-based assessments to facilitate this gold standard science. So those concepts sounds great, but how do you actually get there? And I wanted to give an example of what I mean by evidence-based assessments by using the Institute of Medicine's guidance on conducting systematic reviews. Now, certainly you can't read what's on this slide, but I want to give you an impression of what's involved in a systematic review or an evidence-based assessment and why it has rigor, objectivity, and provides transparency and objectivity in decision-making. So just as I go through, you can see that there are standards for initiating a review or planning the review, for finding and assessing individual studies, using a very structured process. So you're not cherry picking, you're making sure that you have gathered all of the evidence and that you appraise that evidence and assess it in context of your research question. There are then specific methods for synthesizing that information. So how do you do the weight of evidence assessment? How do you bring that information together in context of the decision that you're trying to make or the conclusion that you're trying to achieve? And the evidence-based techniques also require specific standards for reporting. And altogether, just in this example, there are many different frameworks for conducting systematic review, but there are 21 standards and more than 80 elements of performance. So by having this structure to how you do an assessment or how you look at data, how you develop conclusions, adds an additional level of rigor and objectivity and transparency in assessing evidence. I really want to highlight in particular how you bring the evidence together. Based on the research question that was posed at the very beginning, how do you come up with one value or one single conclusion? In my mind, this really is a function of how you bring the evidence that you have together. And there are, again, many different tools. There's structure to help bring this evidence together, both qualitative and quantitative, when you're actually looking at the data themselves. But when you're thinking about the confidence around that and helping to reach consensus, elements such as the validity of the data themselves. Can you rely on the underlying data? Is it a good fit for your purpose? Is it a direct evaluation? Do you have a strength of association? So there are these parameters or factors that can help you characterize your confidence as well as your uncertainty in an underlying evidence base. I would add on this that depending on the research question that you're asking, in addition to assessing confidence, there are other aspects that we talk about with respect to causality or having confidence in relationship that are also important. So thinking about biological plausibility in experiment in particular. And in context of the research question posed, it's really bringing all the different types of evidence together in a weight of evidence to develop your recommendations. I really want to highlight the importance of thinking about uncertainty in practice. I do feel like this is underappreciated and under practice. And to the extent that you can do both qualitative and quantitative characterizations of uncertainty as it relates to your research question, that helps you to inform the potential for consensus and why you may not achieve that. Just very briefly, I wanted to highlight some examples of where I think this has been successful using evidence-based methods. One example is an evaluation where we looked at systematic review of adverse effects of caffeine in a variety of populations. Wanted to highlight that in achieving a consensus or in what we felt like was really contributing to the field was important to have a multidisciplinary team of subject matter experts. So in this example, we had experts from different disciplines, different sectors, informing on, for example, reproductive effects or alertness and different health outcomes, as well as methodologists. So a multidisciplinary team reviewing hundreds and hundreds of papers, so bringing the evidence together in a very structured manner. We believe this helped to update existing beliefs and standards by updating the data or updating the science. Another example I wanted to share was a systematic evaluation. This relates to dioxins and the potential for contaminants in food and how to evaluate that from a risk assessment perspective. I won't go into the detail here, but really wanted to emphasize the methodology. So that began with conducting a systematic review of all of the available evidence. Then assessing the uncertainty or certainty around these estimates. So thinking about a single value, we applied a Bayesian technique to say, what do the data say about that single value and the uncertainty around that? And then by doing those two steps, the World Health Organization adopted the values coming out of that research. So I think that was a good example of how using these evidence-based techniques and characterizing uncertainty helps to inform decision-making and policy. I also wanted to mention that there's kind of building on the evidence-based methods to look at the science. There are also frameworks to help look at that evidence and move to policy. So similar to systematic review, there are several different frameworks available. I'm highlighting just one from GRADE, wherein this gives you a structured approach to go from your evidence to your decision making. And in doing that, there are also more criteria. So similar as I showed you the IOM steps for systematic review, there are criteria to think about how to prioritize some of these decisions, how to integrate the types of findings relative to the policy or the decision you need to make, assessing parameters such as feasibility, your certainty, etc. And one last thought that I felt like it was important to add to our discussion today is thinking about this concept of unbiased and lack of conflict of interest in gold standard science and how that really relates to consensus building. I think it's important to really set the stage that, and I'm talking about bias within individuals and within panels making decisions, not within the data itself, but that really these evidence-based methods and really they call for disclosing different types of bias, and this includes both non-financial and financial bias, which is very important when it comes to trustworthiness and transparency. And then importantly, the goal through these evidence-based methods is to not only disclose, but to manage bias and conflict versus simply saying removing people that might have expertise, but rather to manage that appropriately. And with that, my summary messages to this discussion is that I really feel that evidence-based methods can help facilitate consensus when possible, but certainly convergence. I do put the strike line through just to be a bit provocative, just recognizing that the norms and beliefs will always be present and need to be managed. These methods can really help to inform scientific certainty and weight of evidence. I would also add that really doing a good job assessing your confidence and uncertainty around the scientific data themselves are very critical both to consensus and convergence, and to be able to explain why there might be differing opinions based on the underlying science. These types of techniques, I believe, inform this aspect of reasonable certainty that was introduced at the beginning of the session. And with that, I would like to thank you and looking forward to the discussion.
Thank you, Danielle, for those insightful perspectives. Appreciate you sharing your thoughts. I'd now like to introduce the next panelist with my very American accent, which I hope does not insult, Dr. Berthold Koletzko, who is the Eltz-Kruner Senior Professor of Pediatrics at LMU in Munich. He also currently holds leadership roles in European and international pediatric and nutrition organizations. Bert, I'll turn the floor over to you.
Thank you so much. I'm excited to join this discussion on a really interesting and important question. I'm looking forward to the comments and further discussions in this meeting. So let me just try to share my screen. I thank you, Tricia and Daniele, for making my job easy by introducing the topic so well. But I admit, Tricia, I will deviate from the question you posed, the question to define a single representative concentration of a given component in human milk, because here's a lack of consensus, if you will, because my view is that the key characteristic of human milk is actually its variability. So I chose to rather review approaches of establishing scientific consensus. In terms of my disclosures, I admit that I am biased towards breastfeeding. Our work is financed mostly by public funding, but we do have some funding from companies with an interest in infant feeding. So the concept of consensus is not really a scientific concept. Scientists don't aim for consensus, but they aim for challenging existing knowledge, for raising questions, for trying to refute established knowledge. And so the concept of consensus is not something that is intrinsic to scientific research. But of course, it's important for regulatory decisions, because policymakers, the public, expect consensus on safety and suitability of products and of components of products. And typically, this is based on a review of the scientific evidence and then a consensus on how to interpret the evidence and what conclusions can be drawn because of the huge impact on health and development of the highly vulnerable population of infants and young children. So that's easy to achieve if we have truly convergent evidence, like in this example of a recent meta-analysis, where you can see that a large number of randomized clinical trials, many with high quality, really come to a convergent conclusion. The results are clear, the heterogeneity is limited, the I-square is clearly below 50%, and then it's easy for a group of experts to come to a consensus on the question posed here. However, that's not typical for questions that we address in infant nutrition, because oftentimes we have imperfect or divergent evidence that requires scientific evaluation and judgment. And you see an example here of heterogeneous studies in this meta-analysis. And obviously, for many questions, we would be happy if we had so many questions. And for many questions, we have a limited number of studies. So then the challenge is much greater to come to a consensus conclusion. So what do some regulatory bodies do? I try to review some of those approaches and some more, and what is in common in all of them is clearly to have a review in a systematic fashion of the available evidence. Daniele very nicely described some of the underlying principles. The different agencies have different ways how to apply that, but that's basically the very basis of it. And some have public consultations and others don't. I would think this is a good idea to have a public consultation beyond the panel itself, but that's a question to be discussed. So what do EFSA panels do, as an example? The European Food Safety Authority. A key element for the EFSA panels, and I guess an important question for all such bodies, is that the panels have to be composed of a multidisciplinary group with very different backgrounds, experiences, qualifications, and therefore different perspectives on one and the same topics. And I think this is an essential element for any such reviews. EFSA panels aim for consensus, but consensus is not defined as unanimity. So most panel members should agree on the scientific conclusions, but even if some have reservations, consensus can be concluded. Generally, in EFSA panels, there's no formal voting threshold, no specific percentage defined on agreement that is needed for consensus. The chair facilitates agreement through discussion and refinement of the draft opinion. And importantly, if there is disagreement, such minority views are transparently documented. And I think this is a very important principle, so that people who use the conclusions can review this and interpret this. GRAS panels, similar concept, independent scientists, usually with different backgrounds. And here also unanimity is preferred, but it is not required. And again, dissent must be clearly documented. Codex Alimentarius is an example of a very complicated and winding road towards a consensus. I remember in defining the standard for infant formula many years ago in Codex Alimentarius, it was as fast as 13 years to achieve consensus on the infant formula standard. But it's again the same concept. Consensus is defined as absence of sustained opposition, but not necessarily unanimity, and minority opinions or reservations are then noted in the minutes of the Codex sessions. The European Medicine Agencies, in a different area, deciding on approval of drugs for market introduction, has also a formal procedure for adopting opinions, and consensus is defined if all members, and a quorum is required, agree during the discussion. but also a majority vote can lead to a decision. It can be simple, qualified, or 2/3, depending on the question and the committee. And again, divergent positions are recorded in the final opinion. In medical guidelines in Europe, we have defined criteria for consensus. I show you two examples here for the guidelines of the European Society for Clinical Nutrition and Metabolism, SciPinion, and for the German Working Group of Scientific Medical Societies, where basically all the medical societies in Germany follow their concepts in their guideline development. So for SciPinion, strong consensus is at least 90% agreement of all of the participants of the consensus panel. And the consensus is defined as usually 75% minimum agreement. In the German guidelines, strong consensus is slightly higher, it's 95%. And we try to get there. We aim for the relevant recommendations to go to 95%. If we have a vote below 95%, we go through a second circle and try to accommodate reservations and see whether we can achieve a compromise that is backed up by at least 95%, but also 75% is accepted as consensus. And again, divergent opinions with their scientific reasons can be shared in a comment, in a special vote that is then published with the guideline. So my key message is developing recommendations for medical practice or regulatory decisions. Clearly, we require a thorough and systematic evaluation of the available evidence. Consensus could be agreement of all or almost all qualified experts involved, but it is not defined as unanimous support. There may be deviating opinions of a small group of experts, but they should be transparently reported with their scientific reasoning. Thank you for your attention.
Bert, your observations were incredibly valuable to the conversation. Thank you so much for sharing them. I do want to remind the audience that as you're listening to our presenters, you are welcome to use that Q&A box to go ahead and put some questions in there. And I will take the opportunity now to introduce our next panelist, Dr. John Wallingford, who is currently an independent consultant with Nutrispectives. He was formerly with the FDA and also spent several years in industry. John, I'll hand the mic over to you.
Thanks very much, Danielle. This is Tricia, Danielle, Bert. It's hard to follow Bert in any presentation. Great to be here and hear his remarks and try to address this question about how should a single representative concentration of a component in human milk be chosen. It's a curious question to me. Why is that a curious question? I'm going to give you some examples from groups that have worked on similar questions, looking at assessing safety of bioactive ingredients that might affect the infant's immune system, or that were addressing the quality factors required to be shown for ingredients in new infant formulas in the US, but also to speak to some of the principles that FDA has outlined in their best practices for convening a GRAS panel and their guidance on how they use evidence reviews to determine the strength of scientific support for health claims. I think the key points here will each be explored a little bit. And the first one is understanding the question. The question that was posed to us for discussion, I think, is complicated. And it has a number of different nuances and it has a number of implications. And I'd like to touch on some of those. The second point is that if you're going to try to address the evidence, you need to surround yourself with persons who are smarter than you. I'll give some examples on that. Thanks, Bert, for being here. I'm going to use you as my representative of persons smarter than me for this discussion today. The third one is to dig up the relevant information. And I'm really glad to have Danielle's presentation at the outset. I think this is what she does as a living. And I think that She can speak to some of the processes for identifying and determining the extent of the information that's relevant for the question that's at hand. The fourth one is something that we take for granted as scientists as you're going to think about it. I would like to pose the proposition that you, as a convener of a group trying to reach a consensus on any subject, may have the most direct background and the most interest in that particular subject. And so your thinking is critical, but number 5, getting the smarter people to think about your question is as important. Because the smarter people that have been assembled to help you determine whether there's consensus on a subject have got a zillion other things to think about, and your topic is one of those that they might be able to contribute to, but they're not necessarily going to give it their full weight unless you can find a way to engage them intellectually and tease out what scientific objectives you're trying to reach as something that they are also interested in reaching. The diagram that we saw on going from a body of information towards consensus is a synthetic process. We've heard about some of the procedures for doing that. I'm going to give an example here that's not about the consensus of opinion. It's about the consensus within the science itself. And I think we should look at or scrutinize the science closely to see if whether the science itself is giving us the guide to consensus, not the people that are evaluating it. And the last one is living the scientific method, which is to say that our lifestyle, our beliefs are that science is one of the very few self-correcting areas of human thought, and that scientists by their nature explore what might not be correct in some finding. And so we should always have some humility in any decision about consensus because something else is going to come along. So what are the questions? Understand this question here. What is a single representative concentration for what? Are you trying to establish a database? Are you trying to look for a safety assessment? Are you looking to establish a test concentration for a clinical study? Our question didn't answer those other aspects of what concentration is going to be used for. And who are the stakeholders? Who are you trying to answer the question for? I've got a bias towards thinking about FDA. What is it that FDA is going to do with your statement or your paper? How is that going to fit into their decision making? And are you answering the questions that they need answered? Or are you answering a question that you found was worth answering scientifically, but may or may not help in a regulatory decision? So that's the second part of that. What does success look like? And then I'd like to speak a little bit about the boundaries. What are the terms of reference? Where's the normal state? And how do you decide whether or not any particular concentration is representative? So here looking at the single representative concentration, what are tools that you might look at to see if the sample is right, if the population is right, if the health of the contributors to the sample is right, if the physiological outcomes have been selected appropriately, and whether or not there's evidence of adequacy from other models. We could use a DRI value and look for a 95th centile coverage. In infant nutrition, DRIs for nutrients in the first 6 months of life are almost all established based on mean values from milk of healthy lactating women in usually relatively early lactation, but not uniformly. So we've got sort of this idea that the mean value is going to satisfy the needs most of the time, but that doesn't have to be what you'd use. And certainly disease states should help you define an abnormal situation. If there's some disease that's related to your substance of interest, that would help a lot. And the difference between normal and disease state can be quite profound. So you might be able to get sort of a standard deviation around normal that would still be far from disease state, and that could help you determine safety. I mentioned lactation state in the DRIs. For our purposes, that's an inherent confounder. We've got this idea, if we're adding something to infant formula, that it's going to be consumed throughout the course of the first year of life. And when we know that the concentration of nutrients in human milk can vary twofold or more over that same time period, the lactation state for a sample to find normal, find a single value, is inherently confounded, can't do it. Physiological outcome put forward both mainly in Europe, in Codex, but also in EU, looking at the outcome in breastfed babies is a really important comparator for any outcome of interest in infant nutrition. And that could be at variance with the concentration of a substance to be added to formula, because the bioavailability and the bioactivity of the component may not be the same in formula as it is in human milk. So looking at the physiological outcome is another way of assessing whether or not a single representative concentration can be found. And just to give an example of that, this is a subset of a table from a paper looking at immune factors in human milk in 8 different countries. There were 2 sites in 2 of those countries, so there were 10 total subsamples in this total survey. And I selected out immunoglobulins here because I think that's an area that's of great interest in pediatric nutrition and in infant formula. And I'll just let you peruse here how the mean values, not to mention the interquartile differences, have huge ranges. And you could easily select an IgA value that was a mean in one country that would be far outside the 95th centile confidence interval in another country. And if you selected for just IgA, for example, you could find a value that would be sort of in keeping with the opposite effect in both IgM and IgG. Across these countries, these are, these are rank ordered sort of in increasing amounts. And they're in the same samples, inverse decreasing concentrations for both IgM and IgG. I think one of the things that brings to mind is you might be able to find a concentration that you're after, but you're actually not going to add a pure substance most of the time. And you may need to have specifications around the ingredient that are used to address the companion or the collateral nutrients that are in that ingredient. Now, it's been mentioned a couple times, the objective here of approaching consensus is necessary for purposes in adding ingredients to foods. FDA knows that, and FDA is pretty good at it, actually. You know, they've got the significant scientific agreement standard that was set up at the outset. Tricia, you know that pretty well. Thanks for speaking to that. That is, in its quotes here, high confidence there's a strong consensus in the totality of publicly available scientific evidence. And what FDA did when they established significant scientific agreement is they said, you know, we really want to have this be of such confidence that it's unlikely that our conclusion is going to be revised by new science that comes out after we've made that decision. So again, because scientists are always poking, that may not hold up, and FDA may someday have to even revisit the significant scientific agreement decisions that they've made on the basis of new information that becomes available. And so they have a second category for the qualified health claims that are supported but don't reach significant scientific agreement. And I think that's a really worthwhile thing for us to think about, when that's actually where a lot of the work that is done in pediatric research is at the moment, or someplace between new data and reaching high confidence of a strong consensus. The reasonable certainty standard relies on the minds of competent scientists. I like that a lot. It's a very human acknowledgement that we're going to put this in front of competent scientists and give them a chance to hash it out and learn as best they can whether or not there's a reasonable certainty of no harm of a new substance. And that is the standard that has to apply for GRAS substances as well as for food additives. And FDA has their own application of the grade system, so I won't return to that. Danielle, you can speak to that. FDA also have given guidance on how to convene a GRAS panel. And I've focused on two of the areas that are prominent in their best practices document. One is that you have the right expertise in the room. And one is that there are certain kinds of guidelines or barriers to bias or barriers to non-representativeness that are given consideration and that you've written it down. And here I think, and maybe this can go back to Bert and talk about the EFSA panels that are employed in Europe for similar kinds of decisions as GRAS panels. I'm a little, I know this somewhat, but not as well as I should. My understanding is that there's quite a good number of individuals. You can apply to be on a GRAS panel, and if you meet the requirements, you can be seated. And there can be quite a good number of people on a European EFSA panel. And they can establish a working group below them to do a lot of legwork and then come back to the panel, and then the panel can send their recommendations to the commission. So there's a few levels of examination of data. And in the GRAS model, it's oftentimes a relatively small number of individuals that are asked to be representative of the scientific community. And I think despite the effort to get a balance and breadth of scientific experience on GRAS panels, I think it still is something that can be called into question in the model that we use for GRAS panels compared to the EFSA model. Surrounding yourself with people that are smarter than you. I speak from the experience that we had in convening the panel on effects of bioactives on the immune system, the developing immune system. And while we're looking at this from an infant formula standpoint, and you might want to have a lot of nutritionists, really the question that we were trying to address was the developing immune system. And so it was critical for us to find experts that could speak to exactly that and didn't even necessarily have any experience with nutrition. And finding an expert in the development of Tregs in the intestinal tract was a real important find for us. And finding people that could speak to immune endpoints in samples from around the world to see how representative the US data were from compared to other data sets, that was important for us. And we had an expert in inborn errors of the immune system who could speak to the permutations that occur in disease states and what kind of effects those have on immune system markers. It was really, really important. And the contrast to that, you can have a GRAS panel that has experts who've known each other for a long time and published with each other. And I think that kind of group might be all you got on a new area of science, but it's going to be one that FDA would find suspicious because there's potential for groupthink. And how do you control for that?
Yeah, thanks, John. I don't know if I'm having some technological issues too, but if you don't mind to wrap up here in the next minute or two, we're running a little behind schedule.
Yeah. So this is the digging up of the relevant literature, I think, is where the most fun comes. There's learnings, and as scientists, that's where it's really valuable. It is especially important to identify any contrary points of view so that you can address those. So skipping through a couple slides here in the interest of time, let me give this example that came up in that panel work where we looked at developing immune system markers. And there were 3 different papers that came out within a couple years of each other that all addressed the question of what happens over the course of early infancy to markers related to immune system function. And the top panel was a number of serum metabolome markers and how from the top left corner at entry down to the bottom right corner at 12 months, there was convergence of data in the serum metabolome. And the middle illustration is similar for fecal metabolome, and the bottom was specifically for T cell phenotype. And that these all showed that there were substantial differences at birth or entry into study, but by 6 or 12 months, those differences had vanished. That's convergence in the science that allows for a strong conclusion about the duration of concern and when you might do your measurement markings. And I just come back to my last, the scientific method is what we rely upon. We are one of the few disciplines that employs self-correction, and so go about all this work with some humility.
Thank you so much, John. Your insights were very meaningful. I appreciate it, and very deep. Well, technology alligators got me while you were presenting, so I appreciate the patience behind the scenes. But I will take the opportunity to introduce our final panelist, Dr. Sean Hayes, currently serves as the president of SciPinion, which he also co-founded, and he guides the strategic mission. So Sean, I will hand it over to you.
Thanks, Tricia. So can you see my slides okay?
Yes.
Great. So give you a little bit of background on why I created SciPinion, and then that'll inform how to interpret my conversations and my thoughts. So I've been a toxicology and risk assessment consultant since the mid-90s. And it was in the early 2000s that the activist community started to target bisphenol A. And they were able to get a major retailer like Walmart to ban bisphenol A from water bottles using hazard communication, not risk assessment. And as a risk assessor, I had worked on bisphenol A and I thought it was safe. But what I wanted to know was, what did the rest of the scientific community think? Were we 50-50 or were we 95-5? And from that born the idea for creating SciPinion. Stands for scientific opinion, and that's all we do is determine what the level of consensus is on any given scientific topic. So over the past 8 years that we've been around, we've been trying to figure out how to assemble and conduct these panels of scientists to determine the degree of consensus, and then do it in a way that all decision makers can trust. So I'll give you some of my thoughts about how to go about determining consensus. So some critical questions. If you look at the definition of consensus, is a general agreement, which means somewhere between 51 and 100%. Obviously, 51% is not consensus, and 100% or unanimity is rare in science amongst scientists. And I'd say probably never occurs. There's just always some uncertainty in science. So we're never going to be 100% unanimity. So what is consensus? And I would say that to make decisions, it might be situational, and I'll give you some examples on that. I think it's also important to differentiate between consensus and uncertainty. Danielle talked about knowledge and convergence, and I agree, with greater knowledge, you get greater convergence, but you can still have situations where you have zero uncertainty or very little uncertainty, let's say, and high knowledge, weight of evidence, but still not have consensus. And vice versa, you can still have uncertainty in a scientific situation, but still have consensus on the topic. And there's ways to differentiate the two, and we need to be sure that we do capture that whenever we're trying to measure consensus. So this is just a depiction of two examples of or factors that go into determining the degree of consensus required for making a decision. And that has to do with benefits and risks. If you've got a situation with low risks and high benefits, you probably don't need a high degree of consensus. Whereas if you have a situation with high risk and low benefits, you would want a pretty high consensus to be able to make an informed decision. So we need to keep this in mind when we're trying to decide what level of consensus we want to require. I don't think one size fits all, and it's situational. So talking about the process of how to measure consensus, there's ways of introducing bias in that process, and it has to do with who you ask, how you ask it, and what you ask. So let's go through those. I'm not going to go through all of these because I just don't have enough time, but who you ask has a big factor and can introduce bias in how you measure consensus. I would say that most programs out there that are assembling groups to determine consensus, they can't tell you exactly how they pick their panelists, other than, they want to surround themselves with smart people like John mentioned, which I totally agree with. But how do you define who's the smart people? Is that a subjective process, or can you do it in an objective way? So just in terms of selection of experts, we figured out that, as we all know, we can create a panel to get the answer we want, but that's not the objective. What we want is to determine what's the true measure of consensus amongst the broader scientific community. So what we've developed is a quantitative way to assess a person's level of expertise. We do it in a very transparent way, and it's fully auditable. Someone can go in and see how we pick the panel, and the web app that we created picks the panel for us based on these objective metrics of expertise. And we can also employ a third-party auditor who can come in and score the experts on their expertise levels and use that as part of the criteria for choosing the panelists for a given assignment. So again, building trust into this process all the way through each of these ways that you can go about selecting an expert panel. The how you ask is extremely important. John mentioned the potential for groupthink within a panel of experts, and that is definitely does occur or can occur in many situations. I used to serve on the EPA CAC committee, and I saw a lot of these situations where you had groupthink or deference to who was the perceived expert on the panel. You had dominant personalities trying to sway others on the panel. There's amplification of the first person shared. So what we've created is an online process, not face-to-face. And we use a modified Delphi process, which I'll discuss later. We use a triple blinding process, and we found that to be extremely important. We don't want an expert's opinion to change, whether they're working for company X or FDA or some nonprofit group. We want their opinion to be blinded to that. We also blind the sponsor to who the experts are. We control who gets to pick we control the picking of the experts, not the sponsor. And then lastly, the experts are blinded to each other. This provides psychological safety to the experts. Experts nowadays have gotten attacked in the media for conveying their opinion, and we want to get away from that. So all the experts are blinded to each other. We disclose the experts at the end, but we won't tell you who said what. Again, providing psychological safety throughout the whole process for the experts. One of the other problems I see with how these expert panels are conducted oftentimes is they do the split and conquer the workload so that not all experts are having to do everything. And many times what you see is in panels of one, you might have 10 experts on a panel with 10 charge questions, and they assign each expert to a charge question. And so effectively, you've just got panels of one on each one of those charge questions, We don't allow that. We force each person in the panel to answer their charge questions. We may have sub-panels within our assignment. So a sub-panel on toxicokinetics, one on histopathology, and so on. And they have to answer all of the questions within their subdomain. And lastly, what you ask can bias the process. There can be bias in the questions. They can be leading and frame the answer desired. There can be imbalance in the answer options, which can skew your consensus metrics. Questions don't support clear understanding of opinion. Too many times, charge questions are open-ended that just end up being these text word walls. We advocate for quantitative questions with text explanations so that we can quickly discern what the opinion of the experts are. So our peer review process, again, I mentioned triple blinding. We have what we call a certified peer review process, so that if we have developed this process over time, and if we adhere to it, then we will certify the results. And I'll show you why we have the power or the confidence to be able to say that we certify them later. But again, triple blinding, experts are unaware of the sponsor, sponsors unaware of the experts until the end, and the experts are blinded to each other. We select a panel using objective metrics of expertise, and we use this modified Delphi process, which I'll describe later. And then usually, we're not, if you're the smallest panel we'll run is a panel of 5, and we require a consensus of at least 0.8 in that situation. And that's just a pragmatic situation. With 5 members, what's your minimum level of expertise or consensus that you're willing to accept? But that is definitely situational based on the project. And then our report is full transparency. And like I said, the experts are identified, but we don't tell you who said what. So the modified Delphi process is something we've developed in which round 1 is the independent portion of the panel process. So all experts log in, they download the review material, something like Danielle would put together in terms of a weight of evidence assessment. And then they answer charge questions. And those charge questions deal with the underlying science and the quality of studies, the weight of evidence, and insights on the opinions of the panel. When everyone's done with round 1, that closes. Round 2 opens. Experts get to see each other's answers, and they debate each other. This is the collective learning portion. So they get to learn, hey, I didn't think about that. This expert said that. Can you explain more? So they can debate each other online within the web app. And then round 3 is to answer any clarifying or follow-up questions. And we usually save the bottom line questions, the cancer classification, for instance, questions to the 3rd round. So let them debate the science first and then answer the bottom line questions. And then we can continue with additional rounds if needed, either by design, requested by the panel, because they say they're moving towards convergence. They want another round of debate and voting. So we can always add that into the process very easily. So I said we will certify our results, and that's because we've shown that our approach yields reproducible results. Early on when we were creating SciPinion, we were briefing FDA on our approach. And they asked us, how reproducible is your process? And we said, great question, we'll test it. So we ran 2 panels side by side, 9 experts each, and reviewed the same material, answered the same questions, and they came out almost identical. So statistically indistinguishable. So we have confidence that our approach yields reproducible results. So if we follow our approach, then we'll certify those results. So I just want to talk a little bit about trust in the process. This is really important. Too many times people think that the trust is just in the people that you put on the panel, but really there's other things that are involved. And really transparency is the key. Full documentation of everything, that's the key. And then assuring that you fully capture the dissent and outliers within the panel. You want to be fully transparent about that. But you also want to ask enough demographic questions to better understand why, when there is dissension or outliers. using the triple-blinded modified Delphi process forces interaction and consideration in a protected space for both the majority and the minority on the panel. And we can force follow-up questions to address issues. Again, transparency is key. So I just want to show you some results to consider when you think about trying to undertake this project that INSC is undertaking. And I'll say that confidence in opinion helps to differentiate consensus and uncertainty. So this was a panel we were running on cancer classification of a pesticide, and the expert panel was voting on cancer classification. So we will ask them what classification do they think is most relevant, but we will also ask them in a follow-up question, what's your confidence in that opinion? And so this kind of graph here shows you that confidence, and I'll say that to me, that is hugely powerful and helpful to a decision maker to understand not just, a bar chart on classification 7-1, but understanding this confidence in that opinion. It gets to this level of uncertainty. And there's ways to calculate consensus based on this. An interesting point in this one panel, the one dissenter, 2017, in round one, their confidence was a 3. After they saw everybody else's confidence, they moved it up to a 9. And I'll leave it up to you to decide what was the rationale for that. Hidden sources of bias. Danielle and John and Bert talked about bias in the process. I will contend there's bias amongst all of us. We all have bias. There's no way to eliminate bias from scientists. So this was a project we did in which we were assessing the safety of a compound, and it was based on using omics data. Danielle talked about that being one area where there wasn't high consensus or convergence or weight of evidence. And the sponsor wanted us to get key opinion leaders. They want the gray beards and the gray haired ladies on the panel, but we said, well, let's relax that a little bit. We're a little worried about that. And sure enough, we found a statistical correlation between year of obtaining PhD and inherent confidence in omics approaches. So not too surprising. Those of us who were trained a long time ago, we weren't trained in omics. So we have an inherent lack of confidence in it, whereas those who were trained more recently may have a more inherent confidence in them. So it's important to ask very structured data or structured questions so that you can get to this why. Why are people having this opinion? The charge for INSC is to choose this single concentration. And this was the closest project we've done that gets to that. In this case, we had a client come to us and say, we want help determining the number of doses above which would require child-resistant packaging for an over-the-counter drug. So we thought about this a lot and worked on approaches. And what we came up with is this confidence rating on X-fold therapeutic doses. We assembled the literature, we've developed weight of evidence analysis for the committee to review, but then ultimately it's a subjective decision. And so we asked them, what is your confidence that on a scale of 1 to 10, that a dose of 10x, 20, 30, 40x therapeutic doses is unlikely to cause an adverse effect in a 2-year-old child? And this is for a specific drug, not just in general, but for a specific drug. And prior to this panel, we came up with a mean confidence level of 8 as the criteria above which we would accept that as we would recommend for invoking child-resistant packaging. So in this case, the panel decided that based on the mean responses that only 10x, above 10x doses, it should include or require child-resistant packaging. So you could use this kind of an approach to help identify that single concentration that INSC is after.
Sean,
so in conclusion, yep.
Wrapping it up.
Yep. So that's it. I'll finish with that.
Oh, okay. Yeah. Thank you so much. Yeah, I just want to make sure we have time to get to some audience questions. Great detail, great insights, definitely compelling. And I want to thank everyone again and the entire panel for your presentations. I want to take the last now 15 or 16 minutes to transition over to get a few questions in. We did have some previous questions submitted and had some other questions we wanted to make sure that we get to. So I am going to, several of you touched on this topic of convergence. Actually, I think all of you may have said that in one way, shape, or form. And I do know that there was a recent editorial, which you all are well aware of, that really proposes the term consensus, is maybe restricting our ability as scientists to come to agreement, and that maybe it should be overturned by kind of that convergence idea. So do you think that the scientific community is really being hindered by focusing on the term consensus? Bert, jump in.
Thank you, Tricia. Well, I read the editorial slightly different as you cited it. What is written in the editorial at the very end is, I cite, scientific consensus as convergent evidence rather than scientists' opinion. And that's a different statement than saying replace consensus by convergence. I couldn't agree more with this concept. We don't want just opinions of people. We want a consensus based on convergent evidence, but it's still consensus based on the convergence of evidence. So it's a slight difference in just saying, let's replace consensus by convergence. If we think about the practical aspects, if you think of, say, a grass application, you really want consensus, but again, it's not defined as unanimous decision. It's the majority of experts that come to consensus based on convergent evidence. Sorry for that.
Okay. Does anybody else on the panel have a thought?
I don't know that the scientific community is being hindered by this concept of consensus. I see convergence as being better at conveying the fact that we're moving in a direction. Consensus sounds static, convergence sounds dynamic. And so from that perspective, I'm in favor of convergence, but I don't see a need to have to replace one for the other. I think they each have their own role, and they are what they are.
Tricia, maybe I could add to that.
Yeah, go ahead, John.
In GRASS review, FDA has given guidance on a couple of things, but recent concerns that they've talked about is when there's been unsettled science. And we've sort of struggled with what is FDA trying to say when they describe it as unsettled science. And my understanding of that is that they think that we've not obtained a sufficient body of scientific information. And so no matter how well the experts agree on what's there, what they have to first agree to is that what is there is sufficient. And I don't think that that's always the case.
Okay.
Tricia, maybe I can also add in, I couldn't agree more with the sentiments that have been expressed, and want to actually build on one of the things Sean said, that you can't remove bias. And I completely agree, we're human. And I think that highlights this idea that consensus employs a tier that feels very high, and convergence seems much more evidence-based or science-based. But when you get to consensus, I equate that to almost the differences between science and policy, or science and beliefs. There's other components that go into that consensus decision and that consensus point, kind of call it the highest level. And I think that that's something that's going to be it's very difficult to even characterize. It brings in other aspects beyond the hard science, if you will. We're actually seeing this in the NAM space now. I like the one of the slides that Sean showed, because that is actually one of the things that the field as a whole is trying to address. You've got the science and what can we believe based on the science, but also how do we address the beliefs component, this willingness to accept this new data as part of the overall process for trying to use NAMs in regulatory. So I just offer that as there's a piece that isn't covered by the science when it comes to consensus.
Okay, great. Those are some very interesting thoughts. I'm going to throw another question out there about the threshold. And I think this was talked about a little bit in the presentations as well, but do you think that it is important and helpful to set the threshold to define what consensus would mean? So like greater than 75%, or, you know, like greater than 90% is required. And Bert, I know you talked a little bit about some of the government thresholds that are out there, but I didn't know if that's important to establishing early in the process.
So I very much liked what you, Sean, presented, and I think it's a good point that the level of consensus probably depends on the question. So you probably would ask for a higher level of consensus and greater convergence of the evidence if you look at safety. If you look at suitability, in other words, is it adequate and appropriate, you could probably live with a slightly lower threshold. If you look at benefit, again, a slightly lower threshold than that. So I would say, you know, one size might not fit all. Safety is the most important question for any innovation. So there I would go for a very high level, but for other questions, we could go for a slightly lower level. So I guess that's for regulatory bodies at the end to decide what they want. We in medical societies have our own rules. I showed you what we do in Europe with medical guidelines. And then the consequences are different. If in the medical guidelines there is a 95% strong consensus, then it has a different meaning both for clinicians that read the guidelines as well as for legal issues. If somebody says, you know, you didn't follow guideline, as compared to a consensus of only 70%.
Okay. Anybody else have any thoughts on establishing a threshold?
Yeah, I think I covered that in my presentation.
Okay.
The situation dictates it.
Yep. Okay. Well, I think during the presentations, you know, you raised the concept that defining the question and the objective of the question is critical to gaining consensus. Do you need to have consensus on what the question is before getting to, I don't know, this might be a can of worms. So, but do you need to kind of start at the beginning? What are your thoughts there?
Absolutely.
Yep.
Okay.
On a panel that lost 2 months of work because people had different ideas of what the question was. Yep.
And I would just resounding yes. When we think about evidence-based methods, the first step, the planning, the scoping, well, really the scoping comes even before the plan. Coming up with a research question is incredibly important, and that should be informed not only by your experts but your stakeholders. You know, again, using this idea that you need to have a fit for purpose assessment. So you need to really define the question, have the right people at the table to define the question, as well as setting that, you know, you talked about that threshold up front.
Okay. Okay, so thinking back to, you know, kicking off that very simple question that we posed, and thinking about issues focused on infant nutrition or early life nutrition, how does the level of consensus vary when the experts are academics versus regulators versus professional associations?
Say that again, Tricia.
Okay. So starting back with that early, you know, life question, early life nutrition, how does the level of consensus vary when the experts are academics versus regulators versus a professional association? Or does it?
I guess if I understand the question right, I guess I would answer it with the more important question is, are you asking the right people?
Okay.
You want to have the right experts that are credentialed in making those kinds of decisions. So many times you have like something like you know, a risk management decision that involves, you know, regulatory policy kind of things. It's tough for someone who's only been in the academic world doing toxicology tests to step into that role, and vice versa. So you just want to make sure that you're asking the right experts. And so to me, it doesn't matter what sector they're in, as long as they have the right expertise. That's all that matters.
Maybe I could ask a question to Sean on this. I mean, do those different sectors that Tricia's mentioned have different processes themselves? And my impression would be that a professional society would have a pretty rigorous process of soliciting opinions across the membership that would have a lot of different points of view potentially referenced for the question. So I'm wondering if they have different procedures.
Yeah, that's why I'm trying to make sure I understood the question correctly. So go ahead, Danielle. Do you want to say something?
No, I just said just a small add. I mean, I completely agree with both statements, and it would just add, comes back to having the right stakeholders at the table as well. So not just the right experts, but the right stakeholders to inform that question and know what you need, the types of people, the types of sectors, the types of opinions you need to achieve that consensus.
Okay.
I would say the challenge in professional organizations is that people in the bodies of professional organizations are not always only in these positions because of their scientific expertise, but it's a democratic process. It's majority decisions, and there's a little bit of representation of different groups of societies. So the risk that professional societies can have judgments that are less well evidence-based might be higher than true expert panels.
Yeah. Yeah, and we did get one question that came in early, and it was really about discussing the role that government agencies play in establishing consensus, and when and how that informs guidance and standards. And Bert, I know you touched on that a little bit, in your presentation as well, but does anybody else have thoughts on what role government agencies play in establishing consensus?
That's a slippery slope these days. Well, I'll state this. I don't think the interpretation of science should change with administration.
Fair.
So when you say government agencies, if it's the scientists within the government agencies, then I'm more comfortable. If it's the politicians involved, then I'm less comfortable.
I can maybe share the historic experience from Europe. I was sitting many years ago on the Scientific Committee on Food of this European Commission, which was a body of the European Commission, and basically the administration and the scientific panel were in one hand. And then we had the mad cow disease, BSE crisis in Europe, and there was an outcry that there was not enough independence of the scientific evaluation. It was too much under government control. And that led to the creation of the European Food Safety Authority, with the whole concept that risk assessment and risk management should be clearly separated. Those who do the risk management on behalf of the administration should not do the scientific evaluation of risk and everything aligned with that. And I must say, I like that general concept. Obviously, you still need to talk to each other, but to have an independent risk assessment based on science, independent from the management and political decision, how to then manage it, is, in my view, generally a good idea.
Very well.
I couldn't agree with that more, sitting in a regulatory role. So I just want to say thank you again to the panelists for such a great discussion. And of course, to our audience who submitted some really thought-provoking questions in advance in today. As a reminder, the session was recorded and will be posted to the INSC website in a couple of days. And Cypress, I think you're gonna maybe share a slide with us to make sure that I mention for everybody to sign up for INSC's mailing list and to stay up to date with all of our activities, including the upcoming quarterly update call, October 27th. We also have the next Scientific Roundtable discussion, which will be a discussion defining bioactive, and our tripartite workshop that will be hosted in Washington, D.C. in March of next year. So thank you all for joining us today. Thank you for our wonderful panel of experts who joined us to share their knowledge and wisdom. And we look forward to seeing you the next time. Thank you.
Thank you.